All right, today we're going to dive into one of the coolest shortcuts in all of biology. It's called homology modeling. And what it really does is let us see the invisible. It helps us predict the super complex 3D shapes of proteins, the little machines that run basically everything inside our bodies. And believe me, this isn't just some boring textbook concept. Let's make it real. Imagine a brand new dangerous virus pops up out of nowhere. It's proteins, we know nothing about them. To fight it, we've got to design a drug that can find one of those proteins, lock on, and shut it down. But how do you possibly design a key for a lock you've never ever seen? That's the huge puzzle we're going to tackle. Okay, so to really get the solution, we first have to understand the problem itself. And it's a big one, often called the protein shape problem. So here's the absolute key thing to remember. A protein isn't just, a long string of beads. Nope. It's a complex, folded 3D machine. That specific shape is, and I mean this, everything. It's what creates all the little pockets and grooves that let it do its job, whether that job is digesting your lunch or helping a virus make copies of itself. So no shape, no function. And if you don't know the shape, there's just no way you can design a drug to stop it. And this is where homology modeling swoops in to save the day. Honestly, you can think of it like a brilliant piece of detective work. It's a computer method that basically says, all right, I've never seen this exact protein before, but I'm willing to bet it has a close relative, a cousin, whose structure I do know. It then takes that known structure, which we call the template, and uses it to build a really, really good, educated guess of our mystery protein's shape. So you might be thinking, how on earth is this detective trick even possible? Well, the secret is this amazing shortcut that evolution has handed to us on a silver platter. Okay, here is the fundamental idea that makes the whole thing work. See, over millions and millions of years, the actual sequence, the list of amino acids in a protein, that can change a fair bit. But the overall 3D shape, its fundamental structure, that thing is incredibly stubborn. It changes way, way more slowly. And that, right there, is the big aha moment. Because the structure is so much more stable than the sequence, you can have two proteins from totally different species. They can have different amino acid sequences, but they still fold up into almost the exact same shape. This is what it means for them to be homologous. They share a common evolutionary ancestor. And that family resemblance is what lets us use one to make a model of the other. Okay, so we know the why. Now for the how. How do we actually build this 3D model? Let's walk through the steps. So first things first, that all hinges on finding a good template. The quality of your final model is pretty much totally dependent on what we call sequence identity. That's just a fancy way of asking, how similar is our mystery protein sequence to a known one? You can think of it like this. Get below 30% similarity, and you're kind of flying blind. But once you're in that 30 to 50% range, you can usually build a pretty good model. And if you get above 50%, oh yeah, you can be very confident that your prediction is going to be solid. All right, let's break down the whole process. First up, you've got to go hunting for that template in a huge public database of known protein structures. Then comes a really critical part, carefully lining up your mystery sequence with the template sequence. Step 3 is almost like tracing. You literally copy the main backbone atoms from the template over to your new model. But of course, they're not identical, right? So step 4 is all about building the bits that are different, like the little loops and the sidechains. And then finally, step 5 is all about refinement. The computer goes through the whole model and tidies it up, fixing any atoms that are bumping into each other, making the whole thing as physically realistic as possible. So the computer spits out this beautiful 3D model. We're done, right? Well, not so fast. How do we actually know if it's any good? A prediction is useless if you can't trust it, so checking our work is absolutely essential. And trust me, there are a lot of ways this can go off the rails. Maybe you picked a less than ideal template, or maybe the alignment was a little bit off. Those loop regions are notoriously tricky to get right. Any one of these things can sneak errors into the final model. And that's exactly why scientists have a whole toolbox of clever methods to double-check their own predictions. And one of the heavy hitters in that toolbox is something called a Ramachandran plot. Now, what's cool about this is it doesn't care about the biology. It's a pure physics and geometry check. It just asks a simple question. Is the shape of this model even physically possible? Basically, it looks at all the little bond angles along the protein's backbone and checks if they make sense. You can kind of just think of it like this chart. For a model to be considered good, you want to see more than 90% of its amino acids sitting comfortably in the most allowed zones. If you see a bunch of them popping up in the disallowed region, well, that's a huge red flag. It means you've built a model with parts that are twisted into physically impossible shapes, and you've definitely got to go back and fix something. Okay, let's put it all together now. We've gone from a sequence to a 3D model. So how does this actually lead to real-world medicine? So let's do a quick rundown. The advantages are pretty amazing, right? It's fast, it's relatively cheap, and it gives us vital structural info when doing an actual experiment just isn't an option. But there's one big catch. A major disadvantage. You are 100% dependent on having a known template structure. If your protein has no known relatives in the database, well, you're kind of out of luck with this method. But when it does work, the results can be game-changing. Let's look at a real-world example. A human enzyme called PL2. This thing is a major player in inflammation and even some cancers, which makes it a huge target for new drugs. The only problem? Scientists were having an incredibly tough time figuring out its 3D structure experimentally. So, where did researchers find a template? You're not going to believe this. Bee venom. Turns out the human protein and a similar one in bee venom were 44% identical. Now, if you remember that chart from earlier, 44% is well into the good model territory. That was enough for them to build a solid, reliable 3D model of the human enzyme. And with that model in hand, they could finally start using computers to test which potential drugs might fit perfectly into its active site. And that's how a computer model becomes a roadmap for creating new medicine. And that just leaves us with this one pretty mind-blowing final thought. Every single day, labs around the world are solving new protein structures and adding them to our shared global library. And with every single new template that gets added, our power to model the unknown grows. It's an exponential curve. So it makes you wonder, right? What's next? What currently unsolvable biological mysteries, what diseases that we have no cure for, will we finally be able to unlock because of this?